Okay, so part 2 regarding PBS. Okay. So, evaluation of PBS. Okay. So, this means ano ba yung mga factors na tinitingnan natin using the blood smear. Okay, number 1, under the RBC. So, we look at their size, their shape, their color, arrangement, inclusions, and kung may presence ba ng yung RBCs or yung reticulocytes. Okay? So, yun yung mga tinitingnan natin sa peripheral blood smear. Second, WBC. Okay, so we can check yung total counts, differential counts, IT ratio. IT ratio means immature cells to total count ratio. And then kung meron bang mga abnormal WBCs, mga immature, ganyan. Okay? Next sa platelets, counts din and any abnormalities. Kunyari may immature, mga clumpings, mga giant platelets, mga ganun. Number 4, parasites. Okay, so sa malaria, titingnan natin yung mga in-evaluate natin na yun through blood smear. Next. Okay, so isa-isahin natin yun. Let's start with the RBC morphology. So, erythropoiesis. Okay, so, erythropoiesis is the term, ang term natin for RBC production. Okay. So, general rule, under erythropoiesis, so as cells mature, the overall diameter of the cell decreases, so lumiliit sila. The diameter of the nucleus decreases more rapidly, okay, than does the size of the cell. So, NC ratio also decreases. So, NC ratio is the nucleus to cytoplasm ratio. So, the nuclear chromatin pattern becomes coarser, clumped, and condensed, and then the nucleoli disappears, okay, and then the cytoplasm changes from blue to gray to salmon pink. Okay? Ayan. So, ito yung, sorry, medyo malabo. Okay? Pero ito yung different nomenclatures ng ating maturation stages ng RBC. Okay. So, kung ano yung meron sa isa, ayun lang din yun. Iba-iba lang sila ng term. So, we have 3, normoblastic, rubriblastic, and erythroblastic. Okay? So, ayan. So, for normoblastic, yung first stage natin is the pronormoblast. Sa other nomenclature niya, rubriblast, and then proerythroblast. Ayun, iisa lang yung tinutukoy nun, ibang-iba lang yung name. Okay, so be familiar with all the nomenclatures na lang. Okay, so yun. Okay, so our erythropoiesis start from your megakaryocyte erythroid progenitor. Okay, that will then turn into proerythroblast, early erythroblast, intermediate erythroblast, ayan, and then so on hanggang sa maging RBC siya. Okay, so iron incorporation, okay, starts from the late erythroblast up to the reticulocyte. So, isa-isahin natin yun. So, why do we have to know yung itsura ba ng mga different stages ng cells? Okay? So, that's how we can know alin yung normal sa hindi. Okay? Alin yung normally seen na dapat nakikita natin under our blood films. Yan. So, number 1, we have pronormoblast. Okay, pronormoblast is the first stage ng ating erythropoiesis. So, nucleus niya is round to oval, containing 1 or 2 nucleoli. Purple-red chromatin is open and contains few, if any, fine clumps. So, cytoplasm niya is dark blue because of the concentration of the ribosomes. And then, NC ratio niya is 8:1. Okay. Division, pronormoblast still undergoes mitosis and give rise to 2 daughter pronormoblasts. Then, more than one division is possible before maturation. Location niya is only present in the bone marrow for healthy individuals. And then, length of time is lasts slightly more than 24 hours. So, yan yung itsura ng pronormoblast. So, as you can, as mentioned, normally seen lang siya sa bone marrow on healthy individuals. Now, if you've seen it sa peripheral blood smear, sabihin, hindi healthy si patient natin. Okay? So, ayan yung itsura ng ating pronormoblasts. So, basophilic normoblast, second, nucleus niya is starts to condense na and stains deep purple red. Cytoplasm niya naging deeper, richer blue na siya, and hence the name basophilic. And NC ratio niya is 6:1. So, mas lumiliit na si nucleus. So, undergoes mitosis, giving rise to two daughter cells, and then only in the bone marrow present lang din, and then slightly lasts more than 24 hours siya. Okay? So, this is the basophilic normoblast. As you can see, yung cytoplasm niya mas lumaki na. Okay? So, mas lumiit na si nucleus. Polychromatic normoblast. Okay? So, NC ratio is 4:1. So, hindi ko na iisa-isahin pa yung kasi I believe na-discuss na dito. So, parang pasadahan lang natin. Okay? Then, also not present in the bone, only present in the bone marrow. Yeah. So, polychromatic normoblast, as the name implies, polychromatic. Okay? So, yung kanyang nucleus is mixed in color. So, may dark blue, may pinkish. Yan. So, polychromatic. So, orthochromic normoblast, okay, ang cytoplasm niya nagiging salmon pink na in color. Okay, NC ratio is 1:2, so mas malaki na si cytoplasm kaysa kay nucleus. It is not capable of division na. So, tandaan, starting from orthochromic, hindi na siya nagde-divide. Present only in the bone marrow. Yan, and lasts slightly more than 48 hours. So, that's your orthochromic normoblast. So, polychromatic erythrocyte or reticulocyte. Okay. So, ito na, from here, wala na siyang nucleus, not capable of division. And, ayun. So, as we have discussed earlier, 2 days siya sa bone marrow, 1 day sa peripheral blood smear. Typically, 3 days siyang reticulocyte before it turns into an RBC. Yeah. So, that is your reticulocyte. Okay? So, ini-stain natin using vital stain. And then your erythrocyte. Okay? So, not capable of division, 7 to 8 millimeters in diameter, and then remain active for about 120 days. So, sa normal, mature erythrocytes are biconcave and disc shape and lack a nucleus. So, variations from normal can be classified as variation in red cell distribution, in size, in shape, in color, and RBC inclusions. Now, yan yung isa-isahin natin. Okay? First, variation in red cell distribution. Okay? Number 1, we have the Rulu or the coin stacking or coin pattern, stack coin pattern ng erythrocytes. So, this is due to abnormal or increased plasma proteins. We can also see it in hyperproteinemia, multiple myeloma, Waldenstrom macroglobulinemia, and conditions that produce increased fibrinogen, okay, or chronic inflammation. Okay. So, true Rulu formation is determined in the thin area of the peripheral smear. Okay. Why? Because on the thick part, ang RBCs po doon ay naturally Rulu information. Okay? Kasi nasa thick part ka. Once pumunta ka ng thin part and then nakakita ka pa rin ng Rulu, okay? So, yun yung true Rulu formation. Okay? Next is agglutination. Agglutination or clumping. Okay, mumuo. Okay? So, they have no distinct pattern. Okay? This occurs when erythrocytes are coated with IgM antibodies and complement. So, seen typically in cold autoimmune hemolytic anemia or cold agglutinin disease. Okay. Yeah. So, agglutination naman, sometimes, we can also see agglutination kapag traumatic yung extraction. For example, nahirapan ka, natagalan ka sa fishing, paulit-ulit yung hila mo ng plunger, it causes, napapabilis yung clotting ng ating blood. Okay? And then, bigla ka nakakuha, parang nagkakaroon kasi na ganun yung microclots. Okay? So, best is if in doubt talaga sa extraction. For example, nahirapan ka, kumuha ka na lang ulit. Okay? Para mas sure ka. And then also for chemistry tests, hindi natin tinatanggap yung hemolyzed sample. Okay? For example, pag sinabing hemolyzed, pumutok na yung RBC, kulay pula na si serum. Diba? Best, if tingin mo na ay feeling ko hemolyze to kasi nahirapan ako, natagalan ako sa fishing, ganyan. Best is to re-extract na lang, okay? Para, ayun, sure na maganda yung sample natin. Ganun. So we also have different variations in RBC size. So anisocytosis is the term we use for variation in RBC size. Okay, so it indicates a heterogeneous RBC population. We can correlate this with the RDW or red blood cell distribution width, especially when the RDW exceeds 15%. Okay. We can often see this in patients na post-transfusion, post-treatment for deficiency, presence of two concurrent deficiencies, and intrahepatic sideroblastic anemia. So we have three: normocytes, macrocytes, and microcytes. So normocytes is the normal RBC. Okay. Macrocytes, sabihin mas malaki siya, macro. Okay? So macro is greater than 8 micrometer in diameter siya. And then yung MCV niya or the mean cell volume is greater than 100 FL. Okay? So if you have discussed na yung RBC indices sa lecture, yan. Okay? And then microcytes naman is less than 6 micrometer in diameter siya. Okay? And then MCV is less than 80 FL. So ito yung example ng normal, microcyte, and macrocyte. Okay. So as you can see, ayan, yung mga macrocyte, ayan, yung malalaki. Ayan. In comparison sa mga normal na RBCs. So we have different variations in RBC shape naman. Pag RBC shape, poikilocytosis naman ang term natin. Okay, poikilocytosis. So spherocytes are ball-shaped cells. Okay, so these are small, round, dense RBC with no central pallor. Okay, so also para siyang yung macrocytes kanina. Okay, so almost similar. Yeah. So wala na siyang central pallor, then malaki. You have target cells. Target cells are podocytes or Mexican hat cells. Okay, so this happens with hemoglobin concentrated. If the hemoglobin is concentrated in the center, around the periphery, resembling a target. Okay? So, ayan. So, mukha siyang target cell. And then, we have another one, yung leptocyte. Leptocyte naman is a thinner variant of podocytes. Next, we have elliptocytes. Elliptocytes are cigar-shaped RBCs. Nga po pala, in the thin smear, yung talagang as in, patapos na yung smear, if you view there, usually ang cells po talaga is deformed na siya. Makakita ka ng elliptocytes, mga kung ano-anong shape, doon, that's normal lang po. Hindi siya, talaga, hindi siya, for example, elliptocytes agad. Okay? So, that is only due to the smearing. Okay? Kasi sobrang thin na yung part na yun, parang nade-deform na yung cells. That's why hindi din advisable na dun tayo mag-read. Kaya tayo merong ideal part of the smear kung saan tayo nagre-read. Kasi dun, you can expect na yung makikita mo dun, talagang it presents what your patient status is. Okay? Next, ovalocyte. So as the name implies, oval in shape. Teardrop cell, so mukha siyang tear. Okay, so teardrop or dacryocyte. So, pwede nyo palatandaan dito yung cry. So, teardrop. So, it has a single-pointed extension resembling a teardrop or pear shape. Schistocyte. So, schistocyte or schizocyte are RBC fragments. Okay, so, these are fragmented RBC due to rupture in the peripheral circulation. Okay, so kapag pumutok siya sa loob ng blood. Okay, so yung mga naiiwang fragments, yun yung tinatawag natin schistocytes. Okay. So, fragment of various sizes and shapes are present, often pointed projection. Okay, so yan, deformed na siya. Degmacyte or bite cell. They are usually smaller than normal RBC with a bitten appearance. Okay, parang may bite kinagatan na part. Helmet cell or horn cell or keratocytes. So these are this happens. Sorry. So they are basically a schistocyte with a horn-like projection. So keratocyte is a fragile cell and only remain in the circulation for a few hours. Blister cell or pre-keratocyte. So para siyang keratocyte pero magkadugtong pa yung kanyang hollow part. Okay, so hindi pa siya magkahiwalay. So this resembles a woman's handbag. Echinocyte or burr cells. These are RBC with blunt or pointed short projection that are usually even spaced over the surface of the cell. Okay, so evenly spaced yung kanyang pointed projections. In comparison with acanthocyte or spur cell, meron din siyang pointed projection pero unevenly or irregularly spaced. Next is stomatocyte or mouth cells, RBC with slit-like area of central pallor. Sickle cell naman, there are thin, dense, elongated, RBC pointed at each end, may appear as a crescent shape. So, ito yung sickle cell. Okay, also or another term is drepanocyte. Okay, so nadapa siya kaya siya napitpit. Charot. Yun. Okay, next is variation in RBC color. So we have four in RBC color. They usually represent the amount of hemoglobin na meron yung isang RBC. Okay. So normochromasia is our term sa normal. It has one-third clear central pallor. Hypo naman kapag nakulangan, more than one-third yung clear central pallor mo. Polychromasia naman is if merong variation in hemoglobin content, okay, showing a slight blue tinge when stained. And then hyperchromasia naman is yung wala na siyang central pallor. So nasobrahan sa kulay, hyper. Okay. So yung term na hyperchromasia is no longer used. Ginagamit na natin sa kanya is spirocyte. Okay. So spirocyte and hyperchromic cell are parehas na siya ngayon. Okay. They lack a central pallor. Okay. So, ayan. So, normal RBC, one-third yung clear sa gitna. Hypochromic. Severe hypochromic, ayan. So, halos butas na siyang tuluyan. RBC inclusions. Okay. So, RBC inclusions, as the title implies, inclusions, ito yung mga solid somethings, particles, na nasa loob ng RBC na hindi naman usually dapat nandun. For example, ito, number 1, we have your nucleated RBCs. Okay. So, nucleated RBCs, they are usually orthochromic normoblasts but can appear in any erythrocytic stage of maturation. Okay. So, ibig sabihin, immature RBC. So, hindi sila normally seen on healthy individuals. Okay. So, this indicates bone marrow stimulation or increased erythropoiesis. Okay, so basophilic stippling, okay, so dark blue, purple, fine or coarse punctuate granules distributed throughout the cytoplasm. Ang kanyang distinct characteristic is the blueberry bagel appearance. Okay, so nangyayari to kapag yung RBC has precipitated RNA. Okay? Howell-Jolly bodies. Okay, so Howell-Jolly bodies, they are dark blue, purple, dense round granules, usually one per cell or occasionally multiple. Ito naman is DNA fragments. DNA fragments naman siya. Yung sa RNA, ito, DNA. Heinz bodies. So, using supravital staining, makikita natin yung round, dark blue, purple granules attached to the inner RBC membrane. So, these are not usually seen kapag right stain yung gamit natin. So, dapat supravital stain. So, this is inclusion composed of denatured hemoglobin. Pappenheimer bodies. These are irregular clusters of small light to dark blue granules, usually clumped together near periphery of cell, are called siderotic granules when observed in Prussian blue stain. Ito naman is composed of iron. Cabot rings are thin red-violet, single to multiple ring-like structures that may appear in line or figure 8 shapes. So this one is these are remnants of mitotic spindle. Okay, and then malarial inclusion. So will depend na lang kung anong type of malaria ang meron kay RBC. Hemoglobin H. Okay, so we can view hemoglobin H using supravital stain then. Okay, so they are fine, evenly dispersed, dark blue granules, imparts the Gulf blue appearance to RBCs, so not visible in right staining. Okay, so this is composed of the beta globin chains of hemoglobin B. Okay, hemoglobin C. Hemoglobin C naman are hexagonal crystals of dense hemoglobin formed within the RBC membrane. Hemoglobin SC are finger-like or quartz-like crystals of dense hemoglobin protruding from the RBC membrane. Naligo. Okay, yun. So, we're done with RBC. So, let's proceed with platelets. So, for platelets naman, ito yung ating platelet production. Okay, so we termed it as megakaryopoiesis. So you start from your myeloid stem cell, go to megakaryoblast, and then to megakaryocyte. And then yung platelets are only a part of the megakaryocyte. Okay, fragments ng megakaryocyte, yun yung platelets natin. So hindi sila buong cell. Okay. So yun yung itsura ng platelets natin. There. Ito yung stained, ito yung unstained. Okay? So, yung unstained, ayan, yung mga puting bilog. Okay? So, yung platelets po, mas madali siyang i-view kapag unstained. Why? Kasi, yung alikabok, mga dumi-dumi, nai-stain din kasi siya ng ating stain, and nagmumukha siyang platelets. Okay? So, kapag unstained yung smear like this one, hindi mo makikita yung mga alikabok, yung mga ganyan. Kung ano yung puting makikita mo, yan, this one, ito, sure ka na platelets yun. Okay? One phenomenon regarding platelets na you should remember is the platelet satelitosis. So this happens when there is an adherence of platelets around the neutrophils to produce a ring due to immunologic phenomenon. Okay? So yun. And another thing, wala akong slide, I don't know why, is the giant platelet. So as the name implies, pag lang siya, malaking platelet, yun na yun. Okay. Yeah. And then, balik tayo sa platelet satelitosis pala. Platelet satelitosis also happens in some people kapag sa EDTA tube mo siya kinolect. Okay? So their cells have certain reactions with the anticoagulant na EDTA. Okay? So one thing we can do is diba, kunyari, nira natin siya sa machine, nakita natin mababa. So, ang gagawin nyo is, isi-smear mo siya. Pag smear mo, you've seen platelet, nakakita ka ng platelet satelitosis. Okay? So, what we can do is, re-extract kay patient using citrate tube naman. Okay? And then, citrate tube naman yung gamitin mo to make a smear and then see if magkakaroon pa rin siya ng platelet satelitosis. Yan. So, if wala ng platelet satelitosis, you would know na it's because of the EDTA tube lang. Reaction lang ng blood sa EDTA tube. Okay? And not a immunologic phenomenon. Okay? So that's it for the part